Hey everyone, welcome to another episode of What the Dementia by Bambu Care. I'm your host, Brianna Wilson. I am a certified dementia practitioner and the founder of Bambu Care. So today is a special episode. It was an unplanned episode, not one that I thought I'd be doing. And so I'm not in my normal recording situation. So if you hear odd sounds or if the audio quality sucks, I am sorry, but I felt that I would be doing you all a disservice if I kept quiet about it. And so, though this isn't something that we typically talk about, we need to talk about it, okay? So, if you haven't heard already, there's been a lot of buzz around the potential fabrication and manipulation of images in the Alzheimer's research industry. There was an article that broke on July 21st, I believe, about an influential 2006 paper that was published in Nature, and now some of the study's data and corresponding images are up in question. So this may be a long podcast episode. I honestly don't know because this topic can get pretty deep, but I am going to try my best to reel it in, okay? So if you're interested in this kind of information, which I think it's pretty important, but I strongly encourage you to stick around for the whole shebang, okay? But first, two quick things. So Bambu Care has a free support community for dementia care partners called Bambu Care Champions. So no matter where you are from or what type of dementia your partner may have, all are welcome, and we love to support you along your care journey. So if you're interested in joining the community, please visit letsbamboo.com/champions or check out the podcast notes. Also, we have opened the waitlist for our With Intent Masterclass, which is a comprehensive 9-module course that goes along with our dementia care book, With Intent, a practical guide to navigating behaviors along the dementia care journey. And so early bird pricing will be opening soon, and so if you don't want to miss out, be sure to sign up for the waitlist, and you can check out the podcast notes for that link, okay? All right, so let's get into it. So first, I want to let it be known that fraud in research is nothing new. You just don't hear about it until you hear about it, if that makes sense. And this happens to be one of the times that we are hearing about it. But it happens more than you think and more than people like to admit. And that's a whole other road that we could go down. So let's talk about the research in question and what they were actually looking at, because I think it's important that we have that background, okay? So basically, there's a researcher named Dr. Karen Ash of the University of Minnesota who hired a neuroscientist named Sylvain Lesne to lead a study. So the 2006 article that's creating all this buzz is called A Specific Amyloid Beta Protein Assembly in the Brain Impairs Memory. And it was published March of 2006 in Nature, which is a multidisciplinary science journal that's known for its peer-reviewed research. So in this study, they used TG2576 mice, which are the most well-characterized and widely used mouse models of Alzheimer's disease. And so they're used in Alzheimer's disease research to test therapeutics targeting Alzheimer's disease. And the reason for this is because old mice, which are those that are older than about 14 months old, have a tendency to develop neuropathological features of Alzheimer's, including amyloid plaques, dystrophic neuritis, and inflammatory changes, but without neurofibrillary tangles, significant neuronal loss, or gross atrophy, okay? Now, the article mentions that the TG2576 mice express a human amyloid beta precursor protein called APP, which is supposedly linked to Alzheimer's disease. Their purpose in using these mice was to investigate the cause of memory decline in the absence of neurodegeneration or amyloid beta protein amyloidosis, okay? So what they found is that Mice 6 to 14 months old develop memory deficits because of the accumulation of soluble amyloid beta, termed amyloid beta star 56. And what they did is they purified the amyloid beta star 56 from the brains of these mice and injected it into young rats and noted memory disruption. So what they proposed is that amyloid beta star 56 impairs memory independently of plaques and neuronal loss and may contribute to cognitive deficits associated with Alzheimer's disease. Okay, so let's break this down a little bit further. Now, in this study, they admitted that there was a challenge in analyzing amyloid beta in the brain because of the need to reliably separate specific pools of amyloid beta. But they claimed that they developed a new extraction procedure to separate the proteins accordingly and to be able to search for amyloid beta star molecules in the TG2576 mice that met certain criteria. Okay, cool. So they did all that, which was explained to a degree in the article, and found that the dodecameric assemblies 56 kDa showed the strongest correlation with memory impairment, but not necessarily causation at this point, okay? And they named this assembly the amyloid beta star 56. I know this is a lot, so are you following? Are you keeping up with me, okay? A lot of this nitty-gritty stuff doesn't matter too much, but I just want you to have an idea of what the study was actually looking at, okay? So then after it was identified, they purified amyloid beta star 56 using affinity chromatography, which purifies proteins and protein complexes by disrupting the antigen-antibody interaction, and then size exclusion chromatography, which is a form of gel filtration. Again, none of this really matters, but it kind of does, which we will talk about why later, okay? Now, here's a switch up that happens in the research, so listen up, okay? You may have already caught this from my initial summary, okay? Now, this has nothing to necessarily do with the allegations, but this is why I say that if you're going to go off research, you have to read the whole thing. You can't take slices from it as you please because you may miss something important that can really change the interpretation of the results, okay? So what they wanted to look at specifically was the effects of amyloid beta star 56 on spatial memory in rats, not mice, rats, Long Evans male rats, to be particular, using the Morris water maze. And they used rats instead of mice because they implanted a cannula into the lateral ventricle of the rat's brain and said that chronic cannulae are better tolerated in rats. Now, cannulae are used for intracranial injections, okay? So long story short, they injected some rats with amyloid beta star 56, ran some experiments, made some comparisons, and came to a conclusion based on their findings, which was that amyloid beta star 56 impairs memory in healthy young rats and strongly supports the hypothesis that amyloid beta star 56 is the principal cause of memory decline in middle-aged TG2576 mice. And they are basically saying that their findings set precedent for identifying other star proteins inducing brain dysfunction and suggest that amyloid beta star is a highly specific form of amyloid beta and offers the potential to develop diagnostic methods to detect it in humans with the possibility of targeting amyloid beta star to essentially intervene before permanent structural changes leading to Alzheimer's disease develop. Okay, so that's the gist. A long gist, but that is the gist. Now, I have my own opinions about the study just from what I read, but that's not what I'm here for, okay? I'm here to tell you what's in question and what the uproar is all about. Okay, so boom. In August of 2021, Matthew Schrag, a physician and scientist at Vanderbilt, was paid $18,000 by an attorney who was investigating an experimental drug for Alzheimer's disease called Simufilm by Cassava Sciences, which claimed it improved cognition partly by repairing a protein that can block sticky brain deposits of amyloid beta protein and focuses on amyloid beta 42, which is an important distinction, okay? But anyways, it was believed that some research related to Simufilm may have been fraudulent. And so Schrag ended up identifying images that were apparently altered or duplicated in dozens of journal articles. The attorney then reported many of these discoveries in an FDA petition, and Schrag sent them all to the NIH, or the National Institute of Health, which has invested a lot of money into many of these studies. So what transpires next is a little bit fuzzy, but ultimately, as the story goes, in December of 2021, Schrag ended up visiting PubPeer, which is an online site that allows users to basically discuss and review scientific research after publication. Mind you, this was a totally unrelated venture to all the things that he found with this whole Simufilm thing, okay? But while on this site, he was led to Lesne's papers and found complaints about figures in Lesne's works, and while going down this rabbit hole, essentially ended up flagging figures in 20 of Lesne's papers, 10 of which involved amyloid beta star 56, and one being the 2006 article that has caused the uproar, okay? Largely in part because Lesne is often credited as the discoverer of amyloid beta star 56, and it was that 2006 paper that informed the rest of the research community about it. And since then, he has been awarded in almost $774,000 worth of grants specifically involving amyloid beta star 56. And on top of that, the 2006 paper has been cited in about 2,300 scholarly articles at this point and has seemingly resulted in increased annual NIH monetary support for studies labeled amyloid, oligomer, and Alzheimer's. And so basically it provided a boost in an area that was already being researched, but that there were doubts starting to form, okay? So here lies part of the issue. But it's important to understand that questions about Lesne's work is not new, which we will touch on in just a bit. But to get back on track, what Schrag found is what he considers red flags. He does not claim that he has proved misconduct, saying that it would require access to original, complete, unpublished, and possibly raw numerical data. He's just simply raising concerns based on his findings, okay? So in the 2006 paper, it seemed to contain multiple doctored and duplicated images. And after expressing his concerns, Science, which is published by the American Association for the Advancement of Science, conducted a 6-month investigation, which ended up casting doubts on more than 70 images in Lesne's papers, with some appearing to be blatant evidence of image tampering. And to be honest, it's a mess, okay? But ultimately, it seems that he may have altered the images to better fit whatever the hypothesis was. Okay, now here's the kicker. So again, the uproar is about misleading 16 years of research in the Alzheimer's field, okay? But there are a number of scientists who initially questioned Lesne's claims that oligomers could be analyzed separately inside and outside cells in a mixture of soluble material from frozen or processed brain tissue, saying that it made no biochemical sense. And if it did, then all researchers would be using a similar method. And I believe that this is the same method referred to when I mentioned in the article that they developed a new extraction procedure. So there's that. One Alzheimer's expert, Donna Wilcock, also added that she's doubted studies that claim to use purified amyloid beta star 56 because they tend to be unstable and can convert to other oligomer types spontaneously, along with some other issues that she pointed out. She also noted that several labs have tried and failed to find amyloid beta star 56, but here's the thing, and this is a big problem in and of itself, and it's an issue with the research field in general. But few labs and researchers have actually gone on to publish their findings about their lack of findings because journals are often uninterested in negative results. And researchers also tend to be reluctant to contradict another famous researcher. In fact, another source, Owl's Forum, stated that many Alzheimer's researchers told Owl's Forum, some off the record, that they tried but were unable to replicate the findings of the 2006 paper, and most did not publish those efforts. So really, I ask, who would be at fault here? Now, I have my own opinions, but I'll let you decide that for yourself. I'll just pose the question, okay? And then we have yet another person, a physician and apparent advocate of the amyloid and toxic oligomer hypotheses, who has even cited the 2006 paper at least 13 times in his own papers. And he stated that he could not even find amyloid beta star 56 in human fluids or tissues, and added that, and I quote, "In science, once you publish your data, if it's not readily replicated, then there is real concern that it's not correct or true. There's precious little clear-cut evidence that amyloid star 56 exists, or if it exists, correlates in a reproducible fashion with features of Alzheimer's, even in animal models." So, in this case, it sounds like science should be guiding science, and that doesn't sound like what's been happening, if you understand what I'm saying. So the point is, more than just a 2006 paper would be to blame for the direction of Alzheimer's research. But there's a bigger picture, and so I'll give you a bit of insight into that really quickly. So in an article published by Alzheimer's Research UK, they noted that it's important to stress that this broad body of research is largely unaffected by these specific allegations, even if they turn out to be true. And other researchers have corroborated this as well. It's important to realize that Alzheimer's disease is a complex one, and amyloid is just one of the features that researchers continue to study. Apparently, of 143 drugs currently in clinical trials for Alzheimer's, only 19 of those target amyloid, okay? So when you hear claims that the last 16 years of Alzheimer's drugs were built on data that was deliberately fabricated, and it's referencing this 2006 paper, please take it with a grain of salt. That is a very small picture insight, okay? Even Dr. Karen Ash, who was a part of the 2006 paper in question, criticized this idea that her research singularly guided federal and drug company spending, and explains that there are two classes of amyloid beta proteins called A beta, and that her efforts have focused on one, while drug makers have unsuccessfully targeted the other potential Alzheimer's treatment. And so I personally agree that it is unfair to pin lack of progress in the Alzheimer's research industry on a single research study and article. It's just a bit far-fetched, at least to me, okay? Now, even with all the mess that's unfolded thus far, Dr. Ash expresses that she still has faith in amyloid beta star 56 and is excited about this area of work the study that she did with Lesne in 2006 is not the only study that she has done on it, and so she still feels good about the direction. And it's also important to mention that Dr. Karen Ash herself is not under investigation. She's just being scrutinized for not doing her due diligence in ensuring the data's accuracy as it relates to the alleged manipulated photos. Right now, it's really just Lesne who is under investigation, and not just for the 2006 paper and those images, but like I said, there's been several of his studies that he's produced these images for that they found issues with, okay? So where does this issue lie right now? So besides Schrag reporting his findings to Science, which triggered the 6-month investigation I told you about, he also reported them to NIH, which basically said that any credible complaints will go to the Department of Health and Human Services Office of Research Integrity, or ORI for short, for review, and further investigation by the grantee universities could happen. But something that you have to understand is that this process can take years, and it's typically kept pretty confidential until there's an official misconduct finding and report, okay? So we shall see what comes from it. We shall see what other information will be shared, but just know that a lot of times it is kept confidential outside of the initial allegations, okay? And then Nature has since published a note that says the editors of Nature have been alerted to concerns regarding some of the figures in this paper. Nature is investigating these concerns, and a further editorial response will follow as soon as possible. In the meantime, readers are advised to use caution when using results reported therein. And then the Journal of Neuroscience is following the guidelines provided by the Committee on Publication Ethics to assess concerns related to 5 of Lesne's other papers. And so yeah, with all that being said, basically Lesne is under some real heat and I'm interested to see what's going to come from it all, but again, it may take some time to see that unfold. So there's definitely a lot more to this news break that I could share, but I wanted to at least give you a more thorough view of what's really going on because I've just been seeing a lot of assumptions and misguided opinions, and opinions are great and all are welcome. I have my own for sure, but at some point we have to read and learn what's actually going on, and so, well, that's where I come in. So hopefully you found this podcast episode interesting and informative. Don't forget to join us in the Bambu Care Champion community at letsbamboo.com/champions and sign up for our With Intent Masterclass waitlist so you don't miss out on our early bird special. If anyone has any questions, comments, or future podcast requests, you can send us an email at podcast@whatthedementia.com. Thank you so much for joining us on another episode of What the Dementia by Bambu Care. We look forward to catching you on the next episode. Take care, and until next time, stay strong, care on, and remember, you are not alone. Bambu Care is always here.